Hello, and a very warm welcome to today's Cell and Gene Therapy Insights Live 30 webinar on scalable AAV production in single-use bioreactors. I'm Charlotte Barker, an editor at BioInsights, and joining me today is Dr. John Zamuda, Director of Cell Biology R&D within the Biosciences Division of Thermo Fisher Scientific. After John's presentation, there'll be a short Q&A session.
Thank you, Charlotte, and welcome to all of the attendees today. My name is Jonathan Zamuda, Director of Cell Biology within our Viral Vector Expression Systems at Thermo Fisher Scientific. And today I'll be talking to you about scalable high-titer production of adeno-associated virus in the GIBCO CTS helper-free production system. So for the first part of the presentation, I'll be doing a little bit of a re-review of the key components and performance characteristics of the CTS AAV-MAX expression system. And at first, just a reminder of what is CTS, or Thermo Fisher's Cell Therapy Systems branded products. So the GIBCO Cell Therapy Systems, or CTS products, are designed to enable clinical and commercial cell and gene therapy manufacturing. All CTS reagents are manufactured under cGMP with cell and gene therapy specific intended use statements, extensive safety testing, and proactive regulatory documentation. And all of these measures are taken to provide quality materials to help minimize risk, ease the burden on your quality systems, and support your regulatory submissions. So there are a number of key components that comprise the GIBCO AAV-MAX helper-free production system, beginning with the GIBCO Viral Production Cells 2.0, which is a clonally derived 293F suspension cell line. The GIBCO Viral Production Medium, available both as a liquid and an advanced granulation technology, or AGT format, the AAV-MAX transfection reagent and booster, as well as the GIBCO ViralPlex complexation buffer, and the AAV-MAX enhancer for generating higher AAV titers. Finally, the AAV-MAX lysis buffer, a polysorbate 20 base lysis buffer, is utilized for liberating AAV from the cells at the end of the production run. Some of the key attributes of the AAV-MAX helper-free production system include the ability to generate high titers across multiple serotypes. So utilizing ddPCR to get an accurate read of VG per mil, we typically see greater than 5E10 VG per mil in the unpurified crude supernatant, which makes the system cost-effective. So the higher the titer that you have, the larger the reduction in the production costs for your system. The system offers a scalable, simplified workflow utilizing suspension cells and optimized protocols that are easily ported into different scale production runs. All of the reagents are animal origin free, so we have no animal or human origin components in any of the system reagents. And our key cell line, or VPC 2.0 cell line, is a clonally derived 293F cell that's documented as arising from a single cell and producing very high AAV titers. The AAV-MAX system comes in both RUO and GMP manufactured options, so this can be used for early research all the way through clinical and commercial production of your AAV. So I'll take a few moments just to highlight the GIBCO Viral Production Cells 2.0, which form the foundation of the AAV-MAX system. And as mentioned previously, the VPC 2.0 cell line is a clonally derived cell line from our 293F suspension cells, allowing for superior AAV production. And some of the key attributes of the VPC 2.0 cell line is that the cells have no SV40 large T antigen associated with them. They have not been engineered in any way, but they have been selected for very high titer viral vector production. These cells grow to very high densities, even in routine shake flask cultures, can typically attain 12 to 15 million cells per mil in shake flask cultures. And one of the key attributes of these cells, they have a very non-clumping phenotype, which allows you to essentially grow your cells to the density that you would like to transfect at, and transfect in the same media that you grow the cells in. As I will show later in the presentation, these cells have offered so far very robust scalability. And in the second part of this presentation, we'll be talking about a case study utilizing the VPC 2.0 cell line as part of the AAV-MAX system to generate a clinically relevant AAV at the 50-liter scale. These cells also have a very fast recovery post-thaw, which allows the cells to come out of thaw and be ready to go into your seed train very quickly to save time on the production floor. And similarly, their fast growth, so less than 24-hour doubling time, once again allows these cells to take off from thaw, get into your seed train, and be ready for transfection in as less time as possible. In addition to the RUO VPC 2.0 cells, we also have documented CGMP manufactured VPC 2.0 cells available as part of the CTS AAV-MAX system. So looking over some of the performance characteristics of the AAV-MAX system, and in particular here, the VPC 2.0 cell line that forms the foundation for that system, if we look in the top left-hand side, we see the recovery and the doubling time of the cells coming out of thaw. So across multiple different cell banks, you can see that the time of thaw, the cells rapidly recover at day 0 to day 1, roughly doubling in density, and then really taking off between days 1 and 3, with the viability remaining very high throughout the entire recovery process of the cells. Once again, this is critical to ensure that the cells come out of thaw with high viability, get into your seed train, and allow for rapid scale up to the densities required for transfection. Below that on the left-hand pane is the doubling time of the VPC 2.0 cell line, calculated across multiple different passages ranging from passage 8 to 46. So you can see, again, very consistent doubling times, cell growth parameters across a very broad range of cell passages. In the top middle pane, you can see a representative growth curve obtained through simple shake flask cultures, where the VPC 2.0 cell line is attaining viable cell densities of greater than 15 million cells per mil before the cells finally start to die off at around 7 days post-seeding. In the bottom middle pane, you can see that nice non-clumping phenotype that I had mentioned. So in that shake flask on the right-hand side, those cells are at about 5 million cells per mil, 99% viable. On the left-hand side is a ViCell image showing the very nice non-clumping phenotype of the VPC2 cell line that allows for these cells to easily be transfected in the same media that they're grown in. If we look on the top right pane, you can see between passage 3 and passage 25, so a very broad range of passages. In dark blue is the viral titers obtained for AAV 2, 6, 8, and DJ at passage 3 versus light blue at passage 25. And you can see once again, consistent with the very similar doubling time over a broad range of passages, the cells are also similarly productive for their VG per mil across 4 different serotypes. And if we look at the VPC 2.0 cell line in the bottom right pane compared to the original VPC cell line, you can see that typically we can get between 5 and 20-fold higher titers of AAV utilizing the new VPC 2.0 cell line versus the original VPC cell line. So once again, offering much higher titers and reduced costs in terms of production. Once again, looking across an even broader range of serotypes, we show here the AAV-MAX system generating high AAV titers, again by digital droplet PCR in unpurified crude supernatant across 6 different serotypes, which once again allows us to obtain more viral particles in less volume, and also showing a flexibility to be able to use one system to produce multiple AAV serotypes. And then here we note some of the synergistic interactions of the componentry of the AAV-MAX system and how all of the components, starting from the VPC 2.0 cell line, the viral production medium, the transfection reagent, booster, and enhancers all work together synergistically to give you the highest possible viral titers. And if we look at the bar on the left-hand side, indicating the entire AAV-MAX system, comparing that across various combinations of the VPC2 or VPC cell line with different production mediums, transfection reagents, and enhancers. And again, you can see typically between a 5 to 10 or even higher fold improvement in VG per mil utilizing the entire AAV-MAX system compared to utilizing combinations of cells, different media, and transfection reagents. So really the benefit here of the AAV-MAX system is that it has been optimized fully for all of the componentry to work synergistically with one another to provide the most consistent and highest titers in a highly scalable format. So for the second part of the presentation today, we'd like to share some new data with you regarding the generation of up to 50-liter cultures in single-use bioreactors using an anti-CD19 CAR AAV construct. So for this portion of the presentation, this study comprised generating an anti-CD19 CAR AAV6 serotype from shake flask scale in 125 ml shake flasks up through 5-liter shake flasks, and then taking that same construct into 3-liter and 15-liter thermoscientific high-performance glass bioreactors, and finally into the 50-liter thermoscientific high-performance Dynadrive single-use bioreactor. And in the next slides, you'll see that we've assessed comparability of the cell growth parameters, titers, and metabolites across all of these scales, showing excellent consistency from shake flask to the 50-liter scale. And next, we'll share some of the bioreactor production experimental design, including the streamlined protocol for maximum convenience of scaling up the AAV-MAX system. So typically, we would employ around day -4 prior to transfection of the cells, seeding of the cells at about 0.6 million cells per mil in their bioreactor of choice to grow the cells up to their normal transfection density. Between day -4 and day -1, we assemble and sterilize the bioreactor, and we would get the cells up to the point where we could dilute them to 1.5 million cells per mil at day -1 of transfection in the final transfection volume. On day 0, we would, if the cells had overshot 3 million cells per mil, we would add a little bit of fresh media to get them back to 3 million cells per mil. And then we would add 1% of the AAV-MAX enhancer using a peristaltic pump. Next, we would prepare the plasmid DNA. And here we would dilute the plasmid DNA in cold viral-plex complexation buffer at 10% of the volume to be transfected. So in the case of a 50-liter transfection, we would be making 5 liters of complexation volume. Here, we always add the plasmid DNA to the viral-plex complexation buffer first, as this is very stable sitting out, while we then prepare in step 5 the addition of the booster to our transfection reagent, to the AAV-MAX transfection reagent. And then we complete the complexation in step 6 by simply adding the neat AAV-MAX transfection reagent and booster to the diluted plasmid DNA from step 4. And here, depending on the size of the transfection that you're performing, we would incubate between about 10 to 20 minutes. Typically, a 20-minute time is what we would utilize for a 50-liter or larger culture to ensure maximum complexation, as well as providing enough time to then hook up the complexation and deliver it into your bioreactor. So we would then add the complexation mixture to our cells using a peristaltic pump. And for the different size of reactors, you can see that about 70 mils per minute for 3 to 15-liter reactors and about 1.8 liter per minute for a 50-liter bioreactor. And then in step 8, you see on day 3, cell lysis, AAV harvest, and titer measurement. So at about 72 hours post-transfection, we would perform cell lysis and then measure the AAV titers via ddPCR. And so just to give some example of the robustness of the VPC 2.0 cell line with regard to its seed train and getting up to a 50-liter transfection volume, here all of these cell expansions were performed in our 50-liter thermoscientific high-performance Dynadrive single-use bioreactor that has a 1 to 10 turn-down ratio, allowing us to go right from the 5 liters of cell culture all the way up to the 45-liter final volume prior to transfection. And what you can see here is compared in dark blue is the 50-liter high-performance Dynadrive reactor, and in light blue is a control shake flask. And it's really, truly remarkable how close these profiles match one another from a 125 ml shake flask to a 50-liter single-use bioreactor. As you can see in each step along the way, the cells reach very consistent VCDs prior to being split or inoculated into the larger volumes. And again, at the 45-liter volume, we have excellent cell growth and viability in both vessels. Similarly, we compared shake flask, glass bioreactors, and the single-use bioreactors for viable cell density and viability across the expression run, cell size, and glucose, among other parameters that we'll share in the next slide. But what's evident here is that in the dark red line in each one of these instances is our 50-liter Dynadrive single-use bioreactor. And we're seeing on the left-hand pane very excellent cell growth in the dark red line, and viability, and really across all of the different formats that we showed here, from 125 ml shake flask to 250 ml shake flask, 5-liter shake flask, 3 and 15-liter bioreactors, and the 50-liter DynaDrive. We have very comparable growth kinetics and viability readouts for the cells in each one of these different vessels, making scaling the system very easy from, again, research scales all the way up to clinically relevant production scales. Similarly, cell size tracks quite well across all of the different vessels, as does glucose. And here you'll see a bit of a dip in the dark red line in the high performer reactor, which correlates well with the viable cell density on the far left-hand side. The cells are actually growing a little bit better, a little bit happier in the 50-liter DynaDrive reactor compared to any of the other vessels, and correspondingly you see a little bit of a faster rate of use of glucose between day -4 and day -1 in the 50-liter high performer reactor. Looking at two other key metabolites and byproducts, lactate, you can see once again in the 50-liter reactor, lactate levels tend to stay a little bit lower during the production run out to day 3, but again, well within the range and maintaining fairly low levels of lactate for all of the different vessels tested. Similarly, the ammonia levels. Once again, I think if you look at both the lactate and ammonia at that first portion of the cells being grown up for transfection day -4 to day -1, they're a bit higher, again, because the cell density was a bit higher from the superior growth in the DynaDrive reactors. But then post-transfection, once again, the ammonia levels staying very low in the 50-liter DynaDrive reactor, but again, all well within range and tolerably low levels of ammonia across all of the different vessels. And then to the far right, we can see the anti-CD19 CAR AAV6 titers as determined by ddPCR for a select number of the bioreactors, going from typically our smallest scale 125 ml flask to the 3-liter and 15-liter bioreactors, and finally to the 50-liter DynaDrive single-use bioreactor in the red bar. All of these, once again, were attaining VG per mil of 5E10 or higher. And again, it was great to see that with very minimal optimization, we were able to attain the highest titers in our largest vessel. So going forward, we will be performing even larger transfections to provide direct data and support the AAV-MAX system going all the way up to the 1,000-liter production runs. And we look forward to being able to share that information in upcoming webinars. So next, now that we've made our anti-CD19 CAR AAV in a 50-liter single-use bioreactor, we want to be able to provide also support to move these processes as quickly as possible from research into the clinic. So the final section of the presentation today will be describing a platform for purifying up to 50 liters of supernatant coming from the AAV-MAX system. So for the purposes of this purification, most of the steps here are very consistent with a typical AAV purification. In dark blue on the leftmost chevron, we have approximately 72 hours post-transfection, cell lysis with the AAV-MAX lysis buffer and nuclease treatment all taking place in the stirred tank reactor. Here we're using a Pierce universal nuclease to digest any remaining nucleic acids in the cultures. And in this case, we ran 2 reactors side by side, so you see 2 by 50 liters here. And then we moved on to clarification. And here you can see in the middle of the slide, the depth filtration train that we came up with, utilizing a C0SP, an F0HC, and a Sartapor 2XLG filter train to pass through the supernatant for clarification. And we'll show on the next slide, for the purposes of generating a breakpoint data here, we passed all 100 liters through this depth filter train to ensure that we had accurate ability to clarify 50 liters with some safety margin built in for future purifications. In the particular case of this production run, we also ran a reclarification. So the clarified supernatant was frozen and then shipped for final downstream processing. So upon thaw of that material, we performed a reclarification of 50 liters of the material. And in that case, the reclarification was just run through the C0SP and the Sartapor 2XLG filters to prepare that for concentration by TFF, as shown to the right, using 100 kD molecular weight cutoff, 20x concentration by a 6x volume diafiltration. And then finally to final affinity purification using a thermoscientific porous AAVX affinity resin in a 50 ml column size. So taking a look at the first step of the process, the purification by primary depth filtration, you can see on the right-hand plot that the 3-filter train consisting of the C0SP, F0HC, and Sartapor filters provides excellent clarification of up to the full 100 liters of material that we pass through this filter train. The pressure remained below 4 psi on the primary C0SP filter during clarification of the first 50 liters. So you can see that at the intersection of the orange dotted line and the medium blue line. And the maximum ideal pressure of 10 psi was reached at 250 liters of cell culture per meter squared of C0SP filter area. So we were able to effectively pass approximately 80 liters of the 100 liters through this depth filter train prior to reaching 10 psi. And then at that point, we were able to get up to 97 liters through, the last 17 being a little bit above our target of 10 psi. And here the step yield over this clarification process was a bit greater than 70%. We do expect that the potential to move from the C0SP to a C0HC natural DE filter that we were not able to obtain at the time of this purification might allow us to have an even better step yield over this clarification step in the process. And then as mentioned, because we had to freeze thaw this material, we performed a secondary depth filtration with 2 of the 3 filters from our depth filtration train. Here again, you can see on the plot very low filter pressures for the C0SP and the Sartapor 2 XLG filters, and we had an excellent step yield on reclarification at 91%. So really losing very little in this reclarification process. Typically, one would like to go through this entire process without a freeze-thaw reclarification, but I think these data also show that even in the worst-case scenario where we go through a freeze-thaw reclarify, we're losing very little on that reclarification step. Next, to concentrate the material prior to going on to the AAVX affinity column, we performed, as mentioned, TFF using 100 kD hollow fiber filter that allows the AAV to be retained while the cell media is removed through the permeate. And here the step yield of TFF was 79%. And then lastly, that concentrated and clarified material was loaded onto our 50 ml AAVX affinity column. And what you can see on the plot to the right is that the AAVX affinity resin does an excellent job of selectively binding AAV and allowing all of the contaminating proteins to flow through the column. And that sharp elution peak that you see towards the right with a higher absorbance at A260 than A280 indicates that certainly we have full capsids present within the elution. And here the step yield was really excellent at 97% recovery. So we're getting very efficient clarification, concentration, and finally affinity purification of our AAV6 CD19 CAR construct. And then lastly, if we take a look at the titer in terms of VG per liter, full capsid measurements and infectivity as VG per TU across all of the different steps of the purification train, you can see that we had really excellent recovery of the viral genomes. On the left-hand side, the dark blue bar on the far left is the extracted cell culture, so that's prior to any filtration or clarification. You see a little bit of loss at that clarification product in the first light blue bar. Again, we believe there's optimization that can be continued to take place there to decrease that initial loss that we were seeing. And then to the right of that, the other light blue bar is the reclarification product, the TFF product, and finally the affinity product, where we were obtaining about 45% overall yield from our final product compared to our extracted cell culture. In terms of the percent full capsids, the AAV-MAX system did an excellent job here with regard to percent full. So looking at this, since we did not yet have a full capsid enrichment step built into this protocol, that will be part of the next webinar series that we present on, Here you can see that there's not much of a difference in terms of the percent full capsid as expected from the extracted cell culture through the clarified product, the TFF, and the affinity product. But once again, we're seeing very nice percent full with this construct of approximately 50% full across all of these steps as measured by total capsid ELISA and ddPCR. Lastly, to the far right, we show that this virus also remains infectious. And here from the extracted cell culture on the left through our clarification, TFF, and affinity products, you can see each one of these, we get a little bit better VG per TU for the clarified product, the TFF, and the affinity product compared to the crude supernatant. But once again, indicating that none of these steps are having a significant detrimental impact on the infectivity of the virus, and we're getting very consistent infectivity across all of the different steps of our downstream purification. So with that, I will conclude with our presentation summary. Overall, the data today indicate that the GIBCO CTS AAV-MAX system is a truly scalable solution for clinical grade AAV production. The AAV-MAX system enables highly scalable, high titer production of commercial grade AAV within a very simplified workflow. Today, we've demonstrated scalability of the AAV-MAX system all the way from 125 ml shake flasks through 2 and 10 liter production runs in glass stir tank bioreactors, and all the way up to a 50 liter production run in our high performance DynaDrive single-use bioreactor. And this excellent scalability of the AAV-MAX system enables a truly seamless transition from research to clinical grade and finally commercial production. And lastly, in this presentation, we've demonstrated the purification at the 50 liter scale, utilizing as an affinity capture step the porous AAVX affinity resin to efficiently capture and elute off the AAV construct from these studies. So with that, I will wrap for today. Thank you very much for your attention, and we look forward to your questions.
Wonderful. Thank you very much, John, for that great presentation. So we're going to move on to the Q&A now. Hope you're ready to answer some questions from our audience. So the first question that I've got here, did you have any challenges scaling up lipid-based transfection at the 50 liter scale? And do you expect challenges with the transfection process going into larger scales?
Thank you for the question, Charlotte. With any transfection reagent, there always are unexpected challenges in these processes as you scale up, and that goes for your mixing conditions, your agitation, your temperature, timing, pH. Everything can play a role, and certainly as you go from very small scale to very large scale, those parameters change somewhat. What I would say, though, however, is that our our complexation generation was actually fairly quite straightforward, going all the way from very small scales in terms of deep well plates to shake flasks to wave bioreactors that we didn't show in the data today, all the way up to the 50 liter and in fact ongoing studies, even larger stirred tank volumes. So one has to just keep in mind, again, I think a couple of key steps in that process. One is the temperature of the complexation. So for example, at very small scale complexations, the temperature will actually be increasing as it sits at room temperature during the complexation time, which allows for a little bit faster complexation. So typically we would do, about 10 to 15 minutes at small scale complexation, versus at the larger scale complexations, if you're utilizing cold viral plex complexation media, for example, for these 5 liter or larger complexations, the temperature actually remains quite steady during that time. And so typically there we would extend that complexation to about 20 minutes prior to pumping into the reactors. But generally speaking as well, in terms of delivery of the complex to the different bioreactor scales, the wave bags, shake flasks, et cetera, it's been quite straightforward, very scalable, and we really haven't seen any hiccups even going to these larger scale transfections.
Brilliant. And another question here. What is the difference between research use only and GMP reagents or medium?
So another great question. As I mentioned early on in the presentation, so all of the components of the AAV-MAX system are manufactured under cGMP. However, the CTS branding, our cell therapy systems branding, those products, so the CTS AAV-MAX system, those products are designed to enable clinical and commercial gene therapy manufacturing. And really that's done by the fact that the CTS reagents, while again, all of the reagents are manufactured under cGMP, the CTS reagents have specific intended use statements, extensive safety, stability testing, and proactive regulatory documentation, such that the CTS products will, just allow for the further streamlining of moving a RUO scale process into a clinical and eventually commercial manufacturing process.
Absolutely, and we'll squeeze one more quick question in. What are the recommended run parameters for the VPC cells in a 3-liter and then a 10-liter reactor run?
Oh, another great question. And again, it's been almost, very welcome for us to see that the parameters as we scaled up have remained quite consistent. So if you do go online, there is an application note for a 2-liter AAV-MAX production in a 3-liter stir tank. And it has all of the parameters called out in that application note that you can find on the Thermo Fisher website. And those parameters are highly consistent between the 3 and the 10-liter runs that would take place, for example, in a 15-liter stir tank bioreactor. So please have a look at that application note. I think there's quite detailed information in there. But as always, please feel free to reach out to us, and we'll certainly have one of our upstream experts help guide you in the scale-up process.
Great. Thank you very much, John, for answering those questions. I'm afraid that is all we've got time for today. We are running over a little bit, so we're going to wrap up now. Any questions that we didn't get to, we will try to reply by email. So if we didn't get to your question today, you will still get an answer. The webinar will be available on demand from tomorrow, so look out for an email from us with the link. And I think all that's left is to thank John once more for a great presentation, and thank you to the audience for listening. We hope you'll join us again soon.